I am Nessrine Mezzi. I'm PhD student at Pasteur Institute in Tunis. I'm supervised by Professor Sonia Abdelhak and Dr. Lilia Ramadan. Today I have the pleasure to present to you my work, which is entitled Spectrum of Genetic Diseases in Tunisia, Current Situation and Main Milestones Achieved. Diseases are defined differently from one organization to another. They imply great global burden and a significant challenge for health systems resulting from unmatched patient needs and absence of equal access to diagnosis and treatment. Nearly 6,000 rare diseases have been identified around the world. Only 5 of them have an FDA approved treatment. Approximately 72% of rare diseases are attributed to genetic factors. Genetic diseases are early onset and life-threatening conditions. These diseases particularly concern the Arab world and several African countries. The spread of genetic diseases among these communities, especially rare conditions, were largely explained by socio-cultural factors. Indeed, highest rates of consanguinity with an extreme prevalence of first cousin unions were described across Africa and Arab world. Tunisia is a middle-income African country. It has been inhabited since the Paleolithic period. Berbers, also called Amazigh, are the indigenous people of Tunisia. Through its history, the Tunisian population has witnessed successive invasions and several migratory flows during prehistoric and historic periods, thus shaping its genetic landscape. Similarly to other Arab and African countries, familial and geographic endogamy is a deeply conserved rule in Tunisia with the rates ranging up to 38%. In this study, we reported the preliminary spectrum of genetic diseases in Tunisia. Of public health in Tunisia as their spectrum encompasses 346 genetic disorders. In the present work, we performed systematic reviews of the literature relative to genetic diseases in Tunisia in order to provide a more recent spectrum of these disorders in our population, identify the type of potential health interventions that could be proposed for this condition in Tunisia, and to shed light on challenges that still exist. A comprehensive review of published data was conducted mainly using MESH and PubMed databases. The list of genetic diseases was obtained after querying the MESH database using the keywords genetic diseases. The query results were further combined to additional keywords Tunis, Tunisia, and Tunisian using the PubMed search tool. We focused only on papers published from 2010 to August 2021. Retained papers were included in the meta-analysis and were curated manually. They were completely screened for disease name, OMIM ID, inheritance mode, associated gene, germinal mutations, and epidemiological parameters. We also searched the same information from grey literature. From the selected documents reporting on genetic diseases in Tunisia, a recursive search for additional documents were performed using author names of referring physicians and researchers in Tunisia. Then we used the 10th version of the World Health Organization International Classification of Disease in order to provide a clustering of genetic diseases in different pathological groups. Data have been stored in a local database for subsequent analysis. Let's move to the main results of our study. In this study, we present the actual situation of genetic diseases in Tunisia. Compared to what has been published, 243 additional genetic conditions have been identified. Thus, the total number of genetic diseases in our country amounts to 589 genetic diseases, of which 41.3% come from the present data mining. We opted the International Medical Classification of genetic diseases described in the Tunisian population. Therefore, such classification revealed that congenital malformation, deformations, and chromosomal abnormalities are the most commonly reported. Endocrine, nutritional, and metabolic diseases are the next most frequent group of diseases, followed by diseases of nervous system. Genetic diseases derived from this analysis, autosomal recessive diseases, are the most frequent, followed by autosomal dominant disorders. X-linked conditions were encountered at 5%. 61% of the 589 genetic diseases have a known molecular etiology. Molecular investigation of genetic diseases is mainly conducted using Sanger DNA sequencing in Tunisia. However, since 2013, next-generation sequencing has become more accessible to the scientific Tunisian community, leading to the identification of the molecular basis of at least 45 Mendelian diseases described among Tunisian patients. Target gene sequencing is the main genomic tool used to unravel the genetics underlying rare Mendelian disorders in Tunisia. Heredity is defined as the expression of another disease in addition to the primary disease. This phenomenon has been reported among Tunisian families. In our updated data, we have identified 14 co-occurrences of multiple genetic diseases with a consanguinity history among 6 of them. Both familial and individual comorbidity have been identified. They are either with the same mode of transmission or not. Local phenotypes highlight the genetic disease spectrum among Tunisians. Shannon-Dorfman syndrome, palmoplantar keratoderma, and Parkinson's disease were described in Tunisian patients with unusual presentation. Then, few patients present undiagnosed phenotypes suggesting either new clinical entities or atypical expression of rare or ultra-rare disease. For epidemiological parameters, we focused on the frequency of the whole reported disease spectrum by targeting either the prevalence or the incidence. We noticed the paucity of such epidemiological data, as only 27 genetic diseases described among Tunisians have defined prevalence and/or incidence. For the remaining diseases, only the number of studied patients and/or families is reported. 223 genetic diseases were described to be screened among newborns and/or at-risk individuals in the world, of which 31% belong to the recommended uniform screening panel. Others are diseases that meet the first 6 criteria proposed by AfSHG for expanded career screening panel. Of this disease list, 49% are described among Tunisian patients, representing 19% of genetic disease spectrum in Tunisia, which are therefore potentially target for intervention. Collected data are being integrated into a database under construction that will be a valuable decision-making tool. In this study, we attempted to provide a comprehensive and up-to-date overview of the genetic disease spectrum described among Tunisians. A significant rise in the number of studied disorders were observed in this population. In the period of 11 years, the number of reported genetic diseases increased from 346 to 589. This highlights the availability of better opportunities to perform molecular diagnosis. This suggests also a remarkable progress in Tunisia, particularly through the development of biomedical health research activities, building capacities in this field, and the possibility of international collaborations enabling the use of high-throughput sequencing to overcome the scarcity of NGS platforms in our country. Here in the recent spectrum of genetic diseases. Most of these diseases identified among Tunisians are autosomal recessive. The frequency of autosomal recessive disorders is still high and could be explained by the elevated prevalence of endogamous mating. The clinical heterogeneity could be challenging in molecular diagnosis. Such characteristics render the diagnosis difficult and could lead to misdiagnosis in developing countries such as Tunisia. Screening for founder mutations, especially among endogamous populations, is a cost-effective molecular diagnosis strategy, but it may be inefficient when applied to atypical rare phenotypes and comorbidities. Another impact of elevated rate of consanguinity and inbreeding is the co-occurrence of two or more phenotypes within the same family. Comorbidity, described particularly in inbred populations, makes genetic counseling and prenatal diagnosis challenging. Taking into account the challenge in diagnosis heterogeneous genetic conditions and phenotypes, there are more complex that Another impact of the elevated rate of consanguinity and inbreeding is the co-occurrence of two or more phenotypes within the same family. Comorbidity, described particularly in inbred populations, makes genetic counseling and prenatal diagnosis challenging. Taking into account the challenge in diagnosing genetic conditions and phenotypes that are more complexified by comorbidity in highly inbred populations, NGS seems to be an efficient and rapid genetic diagnostic tool that has marked a shift in genomic medicine in Tunisia. The dearth of data concerning genetic conditions epidemiology in Tunisia suggests that the health burden of such diseases is significantly underestimated in our country. This indicates the necessity of comprehensive epidemiology and patient registry implementation, which could highlight the genetic diseases burden in a population and serve as a worthwhile resource for improving the quality of health care received by patients. From public health perspective, experts and policymakers have opted that newborn and career screening should be conducted as a state-run program. This will allow to an early detection of genetic conditions and congenital disorders, leading to reduced health disparities and less death at a young age. Infant mortality is an indicator of states' health, and reducing this rate is an explicit goal of sustainable development. In addition, reducing genetic conditions expression, particularly those recessively inherited, is a salient issue in several African countries. Targeted screening of prevalent genetic conditions in Tunisia could be a cost-effective alternative in our country, since they have been associated with founder mutations. This could allow to early detect newborns with these conditions and also to identify at-risk individuals. Molecular data availability on relatively frequent genetic rare diseases in inbred populations could be an ideal way that boosts current research focusing on targeted therapies, including gene and mutation-specific therapies, hence leading to the implementation of precision medicine. Several diseases identified in Arab and African communities are subject to targeted therapy based on their genetic information like Bloom syndrome and primary hyperoxaluria type 1. There are several strengths to our study. One of them is that a better knowledge of the genetic disease spectrum in Tunisia is useful for this country as well as for other countries in order to improve the diagnosis, to reduce misdiagnosis, and therefore will have earlier management. There are several strengths to our study. One of them is that a better knowledge of the genetic diseases spectrum in Tunisia is useful for this country as well as for other countries in order to improve the diagnosis, to reduce misdiagnosis, and therefore patients will have earlier management. Molecular and genetic data for these diseases are becoming more widely available through the development of biomedical health research activities and the access to high throughput next generation sequencing. Although the Tunisian population still faces challenges, especially in the case of unidentified phenotypes, which delays the appropriate patient management. These challenges include a paucity of diagnostic facilities, limited access to technological advances. Such issues may be overcome by creating diagnostic referral centers that leverage some technology like high throughput sequencing for genetic diseases elucidation in the country. The specialized structures could potentially offer accurate molecular diagnostic services at a lower cost and allow faster, efficient patient management. Also, patient management should include multidisciplinary team investigators working in a well-defined care pathway. Researchers are needed to accurately set the epidemiological data of genetic diseases, aiming to implement better and appropriate healthcare measures. Patient support groups have a significant role in promoting patient voice and influencing the decision makers. Finally, I would like to express my sincere gratitude to my supervisors, to also to all the members of the project H3 Africa and H3A Bionet, to the African Society of Human Genetics, and to Tanzanian Society of Human Genetics. Thank you for your attention.